Good morning, everyone. My name is Felipe. I am supervised by Dr. Ulisses Rocha at the Helmholtz Center for Environmental Research in Leipzig and by Professor Peter Stadler at the University of Leipzig. And my topic of today is Genome-resolved metagenomics sheds light on the anaerobic conversion of aromatics by complex communities. So, it is known that anaerobic conversion of organic compounds is one of the key points in bioenergy production, and that the discovery of new microorganisms and their enzymes have great value. To allow that discovery, we can use metagenomics coupled with bioinformatics to recover genomes from those metagenomes whose functional potential can be individually investigated. The main goal of this work is to use anaerobic benzene degradation, ABD, as a model to explore functional and genomic aspects of conversion of aromatics. To allow that, we prepared an experiment with a community known of performing anaerobic benzene degradation, and we inoculated bottles with labeled benzene in this community. And what's important here to highlight is that we monitored the activity of the community by measuring the release of labeled CO2, and then we proceed with sampling and DNA sequencing. We then applied bioinformatics to recover MEGs, or metagenome assembled genomes. We assigned taxonomy to those genomes. We determined subspecies by using average nucleotide identity, and we picked the best representative MEGs to perform diversity analysis. And then we did a random forest regression to find the bioindicators for the variable time, which is correlated to the activity of this process. Okay, so as a result, we recovered 2,670 MEGs. We determined 262 subspecies, and we identified 11 bioindicators or MEGs for the variable time. In this figure, we can see the distribution in abundance of our bioindicators over time and among all samples, and we can see clear patterns here. For instance, suggesting our potential key players in the process, and also that there might be synergistic interactions. So in conclusion, we found key players in the process, and there might be synergistic interactions. However, we still need to perform functional notation to draw clearer conclusions, and we are doing this by using ORT suite. So thank you very much for your attention.
Yeah, thank you very much for the presentation. And now it's time for a few questions. I will start with a question. At the DBF set, we are working more in the practical part. I think your research is more fundamental. Do you have an idea when we can use your scientific results for the practical development of biogas plants? Is it in 5 years, 10 years, or in 100 years?
Yeah, sure. I think that one of the most important things here is how this method can be applied in other experiments. Although I'm dealing with a very specific question here in a very specific experiment, we can use this recovery of genomes from metagenomes in many other experiments to know better the mechanisms and which enzymes are important in other processes. So I think that's how I would apply this research here is basically the methodology being defined.
Yeah, from my point of view, it's very important that the specialists in the field of microbiology are working strong together with the engineers. And this is also the reason why we have a strong connection from DBF set to UF set to work with all the specialists together. And I think this is a very good combination. So I see there is no other question right now, but I think afterwards we have also the possibility to discuss some points.